what is the genetic material? So what is the genetic material? Today we know what is DNA, what is RNA, what are proteins, but there was a time when scientists were not knowing that what exactly is the genetic material, right? So although they were knowing that there is something which is present in the chromosomes, but inside the chromosomes also, whether it is the DNA, whether it is protein, or whether it is RNA, what is responsible for being the genetic material, that was a very big question at that time. So protein, now DNA was considered to be as a very boring molecule. Boring molecule in the sense that it has only 4 nucleotide bases, which means they were thinking that how can this be a genetic material, whereas protein, it had 20 amino acids. So they thought genetic material has to be complex. That means protein must be the genetic material. So most of the scientists were supporting this fact that maybe protein is the genetic material, and very few of them were considered that maybe DNA is the genetic material. So therefore, to prove that what exactly is the genetic material, protein, DNA or RNA, there were a set of experiments which were done to prove what is the genetic material. So let's start with the first experiment, that is the Griffith experiment. The search for genetic material. So in 1928, Griffith did an experiment based upon Streptococcus pneumonia. Now Streptococcus pneumonia was a bacteria. You all know it is a bacteria. And what disease does it cause? Pneumonia, right? Simple. Streptococcus pneumonia causes pneumonia. And you will be amazed to know that Frederick Griffith was not trying to find out the genetic material. Instead, he was trying to develop a vaccine for pneumonia. Like these days we are, as you all know, coronavirus is being spread. So we are trying to develop a vaccine for corona. Same way that time pneumonia was creating havoc, and he was trying to develop a vaccine for pneumonia. So instead of that, just by accidental, you can say, discovery, he got discovered that DNA is the genetic material. So these type of accidents are known as serendipity. You must have heard about the penicillin accident also. Anyways, let's come to this. Now here you can see, now what is this diagram? This diagram is a Petri plate. Okay, this is a agar, this is a blood red agar. And here you can see 2 types of bacteria, right? If you observe closely, here in this area, you can see these ones and then these ones. Now, this one, if you observe carefully, and then these ones, both are Streptococcus pneumonia only, but you can see a difference in their structure, right? So the first one, this one was R strain, whereas this one was S strain. You know that bacteria, it can have different strains, right? In biotechnology, we have studied. So R strain, R stands for rough, and S strain stands for smooth. Okay, so there is one rough strain and there is one smooth strain. Now, what is the meaning of rough and smooth? All this information is very much important, because then only we'll be able to understand what exactly the experiment is. Now, rough strain and smooth strain. Now, as you can see, this bacteria, it produces a rough outer edge appearance. This strain of Streptococcus pneumonia produces a rough outer edge appearance, which means that basically it will be like this. Whereas here you can see the S strain, this has got something going around it, right? What is this? This is a polysaccharide. This is a smooth polysaccharide layer, now which makes it virulent. Now here's a new term, that is virulence. Now let's see what is virulence. So S strain is the virulent strain and R strain is non-virulent. Virulent basically means which can cause the disease. So, for example, if we inject the R strain into a mice or a mouse, what will happen? It won't die, as in it will the pneumonia will not be there. If we inject S strain into the mice, then it will die of pneumonia. So virulent means that it is harmful. It can make the mice die, and R strain means it will not make the mice die. Now, this smooth polysaccharide layer, which you can see over here, this is responsible for hampering the immunity of the mice, and that is why the mice dies, because when the S strain is being injected into it. Okay, so this is just a basics of the experiment setup, that we have Streptococcus pneumonia bacteria. Griffith is working with Streptococcus pneumonia bacteria. It has two strains, R and S strain. R is rough strain, which is non-virulent. So if you inject it in the mice, mice will live, nothing will happen because it is a non-virulent strain. Whereas if the mice will be injected by the S strain, the mice is gonna die. So this much is clear. Okay, after that we'll come to the actual experiment, what Griffith did and how he was able to deduce what is the genetic material. So what did Frederick Griffith experiment was actually, what did he do? So firstly, he took the R strain of Streptococcus pneumonia bacteria and he injected it into a mice. So here this is a cute little mouse. Okay, now I'm writing mice because obviously it didn't inject it in just one mouse, it was many mice. Yeah, so R strain was injected into the mice. Now what do you think would be the result? R strain was virulent or non-virulent? Non-virulent. So if it is non-virulent, what should be the result? Mice should live. Okay, so mice lived very nicely. Okay, second setup was he took S strain. S strain is the smooth strain of Streptococcus pneumonia, and again he injected it into the mice, and what did he observe? S strain is a virulent strain, so mice died. Okay, so that was not very shocking because obviously S strain is virulent, so mice will die. Now third, he did a little twist in the experiment. What did he do? He heat killed S strain and then injected into the mice. Now what does heat killed means? Heat killed meaning the, like if you put it in an autoclave and the bacteria is dead. So S strain is dead, as in it cannot produce virulence. That means its virulence power is lost. Now what do you think should have happened? S strain heat killed, that means it cannot give pneumonia. So mice lived. Okay, so that these were his results, what he got. Now in the last, what he did, he took heat killed S strain plus R strain bacteria and then injected it into the mice. Now just try to guess what should have been the result. Heat killed S strain, that means the virulence power is lost. But R strain is also non-virulent. So basically we have a non-virulent strain and the other is also non-virulent. So what should have been happened to the mouse? By normal logic, if you see, heat killed S strain, R strain. So R strain is non-virulent, heat killed S strain, that means you've killed its virulence. So mouse or mice should have been lived. But to his surprise, what happened? He was shocked by this, result that all the mice died. Okay, so this was his experiment. Now, how does he explain this? That if you're taking non-virulent strain, then how did the mice die, right? So what he said was that something was present in the S strain, even if you have heat killed it, that was able to transform the R strain into a virulent form. That means R strain got virulent. It was able to cause pneumonia in the mouse, and that's why it died. So what was that transforming principle? He still didn't know during that point of time. So he tried to deduce it, and what he thought was that there is something which is changing, which is inducing some change in the R strain and making it virulent. But what was that? He was not clear about it. And not only that, when he took the R strain, when he took these dead mice, what did he observe? That the bacteria which he took from there, that was S strain. Remember, we have already added heat killed S strain. So that was very much shocking for him, and he thought that he said that there must be some transforming agent which is transforming R into S strain. So this was known as the transforming principle of the Frederick Griffith experiment. Now, because he was not able to prove what was that which is causing this change, so the next set of scientists came and did an experiment regarding this, in trying to prove what actually the genetic material is, right? Now here I would like to explain one more very interesting thing, which is that although he was not knowing that what has caused this shocking results, but how do you, now that you know that how it is happening, can you deduce that what has happened in the fourth situation? How did the mice die in the fourth situation? Think about it. Now all of you know that in a DNA is double-stranded. Now if you heat DNA, you know that he has used heat killed S strain. So when he heated it, what will happen? The DNA strands are going to separate out. Remember, it will not be cleaved. DNA will be cleaved when we use DNases. Now here we are not using DNases, we are using heat. So because of heat, only the DNA denaturation, this step is known as DNA denaturation, which you must have studied in biotechnology. We'll study it in more detail. So this is separated, DNA is denatured. Now when proper temperature again will be given, let's say 37 degrees Celsius or 25 degrees Celsius, then again it will form a double-stranded DNA. Okay, so this is not given in NCERT. This is not explained anywhere as to how actually this point happened. So heat killed S strain bacteria, its DNA got separated, that is denaturation happened, and when normal temperature, when favorable temperature was given inside the organism, inside this mice, because it has R strain, so this R strain bacteria took up the S strain DNA, and then it caused virulence and eventually the death of the mice. Okay, so now this is clear. Okay, in the next video we'll study about the next set of scientists, what experiment they have done to prove what is the genetic material. Okay.